All right, welcome back to Chapter 10. This is the second part. We're going to be talking about light and wavelengths of light and how that can be used to generate ATP. Once again, this occurs in the chloroplast, specifically in the thylakoids. That's where the light is captured by chlorophyll. Okay, so the colors of the rainbow and sunlight. Light is a form of electromagnetic energy, also called electromagnetic radiation. Like other electromagnetic energy, light travels in rhythmic waves. These form wavelengths. A wavelength is the distance between crests, and we can see a crest right here, which is the top of a peak. A wavelength is the distance from the top of one peak to the top of the next peak. Wavelength determines the type of electromagnetic energy. We can see up here that we even have radio waves and television waves. These are traveling through the air, whizzing right by your head right now. Microwaves used for cooking, infrared waves, X-rays, gamma rays, they all travel through the air in wavelengths. In fact, if you can think of your favorite radio station, like 97.9 or whatever, those wavelengths are traveling at 97.9 nanometers. So it's the direct correlation right there between the radio station and the wavelength. So the electromagnetic spectrum is the entire range of electromagnetic energy or radiation. Visible light consists of wavelengths, including those that drive photosynthesis, that produce the colors that we can see. Light also behaves as it consists of discrete particles called photons. Those photons are relatively important, actually. But it's these differences in wavelengths which is allowing us to actually separate these colors. As we can see, the color red and dark reds have a very large wavelength compared at least to the deeper purples. So 400 nanometer wavelengths ranging up to upwards of 750 nanometers. So a significant difference. And then we have all the colors of the rainbow in between. Taking another look at it, we just kind of flipped it upside down. So purple's on this side, red's on this side, and shorter wavelengths, as we move this way, they get larger and larger and larger, allowing us to separate things. You can almost think of this as sort of similar to density, I suppose. So we'll get more towards why these colors are a big deal in a couple slides from now. But what we want to start looking at now are pigments, the chlorophyll within these thylakoids. Pigments are substances that absorb this visible light. Different pigments absorb different wavelengths. Wavelengths that are not absorbed are reflected or transmitted. Leaves appear green in part because of chlorophyll, but also in part because chlorophyll does not absorb the color green. So therefore, as you can see, pew, it gets reflected. If you look at your neighbor's shirt and it happens to be yellow, well, their shirt has pigments that absorb red, green, blue, violet, and all the colors of the rainbow, but it doesn't have a pigment that absorbs yellow. Because yellow is not absorbed by the pigments in your neighbor's shirt, yellow would pew, be reflected. The reflected yellow wavelengths would then ricochet off the shirt, bounce off it, and it would travel back to your eyeball, and you would interpret the color yellow because that's the only wavelength that's entering your eye as it's bouncing off his shirt. We can measure how much of various light wavelengths are being absorbed by pigments using what is called a spectrophotometer. It's a machine that we can send light through, and we can measure how much of that light is actually picked up by a pigment. And right here we can kind of see how that's happening. We're shooting light through a filter right here. Looks like this filter is only allowing green light to pass through. So light going through a prism, separating into colors, only green light can pass through. Green light is then going through a chlorophyll-filled solution into a little sensor, I suppose, and we can see that there is a very low absorption of that green light. So on the high end here, 100, that's indicating that green light is almost not absorbed at all, because this is getting a lot of green light going through it. So green light almost goes straight through. Boom, green light's picked up. A high amount of green light is sensed by the sensor and recorded. Conversely, looking down here, same process, different filter. It filters out just the blue light. So blue light is going through the chlorophyll, and as you can see, we have a very light line here, indicating in this image anyways that there's very little blue light left because it all got picked up by the chlorophyll. The sensor here would pick up that there's not a lot of blue light, very low reading on our transmitter here, and this kind of helps us understand, or has shown us in the past, why exactly or how exactly or what colors exactly chlorophyll is picking up out of the light. And you can change these filters here to be really any color that you want. So now we're looking at what we were just looking at with the spectrophotometer, but the results of a test conducted by Theodore Engelmann. But before we go any further on that, we should talk about the two different types of chlorophyll, as well as carotenoids, and we can see each of those in this top graph here. Chlorophyll A and chlorophyll B, they're very similar in structure, slightly different. Carotenoids, also we see up here, these are three different pigments that plants have. Chlorophyll A is the main photosynthetic pigment. Accessory pigments would be chlorophyll B, as well as carotenoids. So these accessory pigments are more or less used to broaden the range of light that can be absorbed by plants, by chlorophyll. So we can see right here, if it was just chlorophyll A, plants would absorb this particular wavelength great, and they would absorb this particular wavelength of light great, but not much in between. And that's kind of the necessity of these accessory pigments, is they not allow just this specifically to be absorbed, but it also adds more down on this side, and it extends the range out a little bit this way, as well as a little bit down here. Not much, I guess, but a little bit. And we notice that right in here, there's still not a whole lot that's absorbed. Once again, keep in mind, if it's not absorbed, it's reflected, and green is the lowest line on here. You can't really quite tell so much. I guess down here you can. Green is the lowest amount that's absorbed, so since it's not absorbed, it's reflected or given off. So what's the difference between these top two graphs here? The top one is called the absorption spectra, and the bottom one down here is called the action spectrum. The absorption spectrum is a graph that plots a pigment's light absorption compared to its wavelength, and we can see those wavelengths down here. Light absorption is measured on our y-axis. The absorption spectrum of chlorophyll A suggests that violet, blue, and red light work best for photosynthesis. An action spectrum down here profiles the relative effectiveness of different wavelengths of radiation in driving a process, hence the word action, process, action. The action spectrum of photosynthesis was first demonstrated by Engelmann. In his experiment, he exposed different segments of filamentous algae to different wavelengths, maybe similar to what we were looking at with the spectrophotometers in the previous slide. Well, what he was able to do is he noticed that areas of wavelengths favorable to photosynthesis produced excess amounts of oxygen. And we know that plants give off oxygen, so it sort of makes sense that the more photosynthesis we have going on, the more oxygen that we're going to have. We looked at, in our first video, oxygen being a waste product of photosynthesis. I'm actually going to go back to that in just a second here. Here we go. And we can see oxygen coming out right down here. So going back to the slide we just left, we can see a little bit of his experiment continued down here, Engelmann's experiment. How was he measuring the oxygen that was produced? Well, he's actually using an aerobic bacteria, bacteria that require oxygen in order to function. So you can determine the metabolic activity of these bacteria based on the quantities of the oxygen. So we had previously spoke about chlorophyll A and chlorophyll B. Chlorophyll B was an accessory pigment. Chlorophyll A was the primary pigment, meaning that it does most of the absorption. And we can see a very slight difference in the structure between these two pigments. It's actually right up here. It's CH3 in chlorophyll A and CHO in chlorophyll B. This difference causes the two pigments to absorb at slightly different wavelengths within the red and blue parts of the spectrum. So now that we spoke about light and wavelengths and chlorophyll and pigments, let's look at a little bit about electrons. When a pigment absorbs light, electrons go from a ground state to an excited state, and the excited state is relatively unstable, has a lot more energy. When excited electrons fall back to a ground state, as they all do, they gradually lose their energy. Photons are given off, or an afterglow called fluorescence, which we actually see over here. If illuminated, an isolated solution of chlorophyll will fluoresce, giving off both light and heat. This ability of electrons is what we are able to, well, not we, plants are able to use in their production of sugars, capturing photons from the light, exciting these electrons, and then getting some products released off of the electrons as they calm down, so to speak. But something to consider is when these electrons get excited and all antsy, we need something to kind of hold them and kind of tolerate them, if you will, for a little bit. And that's where these primary electron acceptors come into play. They will accept that excited electron from chlorophyll A and kind of hold on to it. We'll then transfer that electron along the way and release some energy from it. Now, the way that plants can do this is by using what is called a photosystem. And a photosystem consists of a reaction center complex, which we see right in the middle here. We can see the electron going up to it. And this is a protein, and it's surrounded by light harvesting molecules, which we see right here, light harvesting molecules, the complexes. So the light harvesting complexes, which are pigment molecules bound to proteins, they kind of channel or funnel the energy of the photons from the light to the reaction center. So we can kind of see this kind of playing Plinko, it looks like. Boom, boom. And we have an excited electron that's kind of captured by the primary electron acceptor. Running out of time here on this one, so we'll pick up with photosystems as we move into part 3 of chapter 10 here.